Okay, welcome, my dear friends. Regarding of our CPGET free online coaching 2025, this is Dr. Padala Tirupati, Assistant Professor, Department of Botany, SRR Government Arts and Science College, Autonomous Karimnagar. As a convener of this entrance program, free coaching program, today's our eminent resource person, Dr. Gogikar Bhagirath Garu, is an extensively experienced personality in the field of molecular biology, especially genetics. So in this regard, we would like to invite Dr. Gogikar Bhagirath Garu. Please welcome and share your screen and start your session. Please welcome, sir.
Okay, shall I share my screen and good evening, participants. I think everybody seeing this screen.
Yeah.
Visible?
Please click on slideshow, sir.
Okay. Wow. Okay. So in the last class, we discussed about linkage and crossing over phenomena. Yes?
Please click on slideshow, sir.
Yes, sir. I'm already full slide it is.
ఓకే బట్ ఫుల్ స్లైడ్ ఉంటే దాని అర్థం ఏంటంటే ఇది కావట్లేదు అనమాట. మీరు ఒకటే స్లైడ్ దగ్గర ఉంటారు. మీరు మళ్ళీ ఆఫ్ స్లైడ్కి రండి, అంటే స్లైడ్ షో దగ్గరికి వెళ్ళి వెనకకి రండి.
ఓకే.
ఇక అలాగే స్టార్ట్ చేయండి, అలాగే కంటిన్యూ చేయండి.
ఓకే సార్.
రైట్, ప్రొసీడ్.
So in the yesterday we discussed about the linkage and crossing over phenomena. How linkage phenomena is related to the crossing over, and how do we predict different recombinants in the progeny when we cross made between the two plants with two type of characteristics. So once we can observe that when two or more genes located in the same chromosome, we can call as linked genes. So these linked genes commonly they are joined and they inherited togetherly into the gametes as well as next generations. But during the cell division process, these linked genes are separated due to the crossing over. We already known that one. The crossing over is one of the phenomena takes place during the sexual reproduction. When gametes are formed, at the time of gamete formation, during a pair of homologous chromosomes, in this pair, two non-sister chromatids are appeared in X-shaped structures that is known as chiasma or chiasmata. In this chiasma region, two non-sister chromatids are break down and reunion with ligase enzymes and alter the linked genes. So we already discussed the linkage and what is the linkage strength when compared to the distance between the genes. When the two genes are very close together, the strength of the linkage is more and very strong linkage is there in between the two genes. In the same way, the wider genes are there, far away genes are having very less or we can call as a weak strength between the two genes which are far away on the same chromosome. So today, what is this class focus on 3-point test cross. What is meant by 2-point test cross and 3-point test cross also we can discuss it in the last class. So today I am discussing only 3-point test cross. When go for the linkage phenomena, then crossing over phenomena. So what is the linkage map or crossing over maps or genetical maps? Generally, the crossing over genetical maps or the linkage maps indicates first time these maps are discovered by Alfred Sturtevant, first time in Drosophila melanogaster. By using chi-square test, he designed the chromosomal maps or the we can call as genetical maps also. So where is this here? And I will see that how it is, just to go through the diagram. Okay. So once we can draw any homologous chromosome pair. These two homologous chromosome pairs are there. How the genes are arranged on this chromosome? Number of genes are present on the chromosome. What is the distance between the two genes also we can shown in a graphical model or graphical representation of the linked genes on the chromosome, which indicates what information revealed by the chromosomal maps. These chromosomal maps are useful to construct the genetical maps of any organism. How many linked genes are there? What is the distance between these two genes? And also order of the genes also very important. For example, A gene is here and B gene is here. So then X gene is here. So the order of the genes are very, very important. And also we know the correct gene position on the chromosome also. So for this purpose, we can construct these genetical maps in the hybridization process or in the plant breeding or animal breeding process. So this is the usage of linkage maps or genetical maps. First time it is designed or discovered by Alfred Sturtevant in Drosophila melanogaster. Later, so many organisms, this type of genetical maps are constructed and which reveal more information about the genes. Today, in our class, I try to focus on that. What is meant by 3-point test cross? What is meant by interference? And what is meant by coincidence of coefficient? Such type of terminology, and as well as I explain about genetical maps 3-point test cross problems. So first of all, I go through that the 3-point test cross. It indicate that the crossing over takes place with reference to 3 points. With reference to 3 points. That means, for example, I will draw here 3-point test cross is. Oh, 1 minute. Yeah. So this is the 2 homologous chromosomes. So this is the one with reference to 1 point. This is another point on the chromosome with reference to. So if reference to 2 points, so both the points, the chromatids are break down and exchange with each other. In this way, the order of the genes are changed in the gametes. Once it is complete linkage, what type of progeny we are expected? If it is a incomplete linkage, what type of progeny are we expected? Based on that, we can explain how it is complete linkage and how it is incomplete linkage. So I will go through this third slide and we can easily explain that process. Yeah. Complete linkage means what? Only we can expect parental characters in the progeny. We never expect any recombinant types. In this type linkage, genes are very closely associated and tend to transmit togetherly upon coming from the same parent for 2 or more generations in a continuous and regular fashion. For example, the genes for bent wings, BT, the small BT is, the character is bent wings, vangina rekkal anamata. Next, SVN, bristles, Shevan bristles of the 4th chromosome mutant of Drosophila melanogaster shows complete linkage. In Drosophila melanogaster, one of the character which represented by genes are shown complete linkage, 100% complete linkage. See here, this is the normal fly and bent Shevan. So normal fly containing BT plus and SVN plus. This is the homozygous and dominant type, and this is a homozygous recessive type. In the gametes, we receive only one, that is BT plus and SVN plus. Here BT and normal SVN. So F1 gametes are formed, then it combined with each other and it form heterozygous condition. Heterozygous condition seen here. This is the heterozygous condition seen here. So when it is test crossed, not back cross, test crossed with recessive homozygous, we are getting this type of gametes. These gametes united randomly and give F2 progeny. All are with the same type of parental characters. Only two type of characters, wild type and bent Shevan. That indicates what? No recombinant takes place. No crossing over takes place between these two genes. If any crossing over takes place, that is single crossover అంటాం మనం. ఇక్కడ జరిగితే ఒకవేళ this BT నార్మల్ బీటీ అనేది ఈ ప్లేస్ లోకి వెళ్తుంది, బీటీ ప్లస్ అనేది ఈ ప్లేస్ లోకి వస్తుంది. ఇఫ్ క్రాసింగ్ ఓవర్ అక్కర్స్, బట్ నాట్ ఎనీ క్రాసింగ్ ఓవర్, నో క్రాసింగ్ ఓవర్ టేక్స్ ప్లేస్ in between these two genes. That's why it is completely linked genes. No separation of the linked genes. 100% linkage is there. That's why it is called complete linkage. When any two genes are linked and shown same kind of features in the next progeny, without any recombinants, without any formation of recombinants, that indicates what? 100% complete linkage. Parental combination is restored and complete absence of independent assortment. For example, according to the Mendel, in the F2 progeny, we can see along with parental combination, non-parental combination also. That's why here no independent of assortment also. Complete absence of independent assortment. If you go for the test cross in the dihybrid, 1:1:1:1. But only out of the 4, 2 are related to the parents. Remaining 2 are different from parental types. That's why there is an independent of assortment is there according to the Mendel. But this linkage shows that 100% parental combination is restored in the F2 progeny, as well as complete absence of independent assortment. I think it is clear. Next, incomplete linkage. What is meant by incomplete linkage? In this type of linkage, genes which are widely located in the chromosome and have chances of separation by crossing over. If the genes are a little bit wider when comparatively previous one, there is a chance of crossing over is there. Definitely, if crossing over occurs, the parental combinations is also we can expect recombinant types also. Also, we can expect heterozygous cross with homozygous recessive. That is nothing but test cross. If crossing over occurs, what happened? What type of gametes we are expected? If not crossing over occurs, only 2 type of gametes we are expecting. So 2 type of gametes are which related to the parent, and remaining 2 are dissimilar with parental type. For example, B plus VG plus, gray long, it is a completely parental type. And BVG, and this is also homozygous, black vestigial. But see here, B plus and B plus here, VG plus and VG, somewhat different we can see here. That's why in the crossing over, we can see this is the recombinant gametes. Recombinant gametes లో ఒకటే ప్లస్ ఉంది, ఇంకొకటి ప్లస్ లేదు చూడండి, బి ప్లస్ విజి. ఇక్కడ బి విజి ప్లస్ అంటే ద ఎక్స్చేంజ్ విత్ ఈచ్ అదర్, అంటే క్రాసింగ్ ఓవర్ జరిగిందన్నమాట. ఇక్కడ క్రాసింగ్ ఓవర్ జరగడం వల్ల we can get recombinant gametes. So that's why we can say non-parental characters. Gray vestigial is a non-parental character and black long also non-parental character. But always in the incomplete linkage, the recombinants are formed less than 50% only. It not exceed the 50%. That's simply blindly every student remember that. If the two genes arranged on the same chromosome which exhibit incomplete linkage, that indicates what? In the F2 progeny, parental characters are more than 50%. Recombinant combinations or characters are less than 50%. Based on that, we can easily expect crossing over is occur or not. That indicates incomplete language. How can say the distance between these two genes? The distance between the two genes we can easily find out with one of the formula that is equal to recombinant types by total progeny into 100. What is that? The distance between the two genes, that means the distance between B plus and VG plus, we can easily calculate it by one formula that is equal to recombinant progeny by total progeny into 100. We can easily calculate the recombinant percentage in between the two genes. The percentage itself indicates the distance between the two genes, which are expressed in the centimorgans or map units. For example, I will go through this problem only. It is not coming it. Yeah. See here. So number of recombinants is there. For example, 9. We can say 18. 18 we can write here. Divided by total number of progeny. 41, 41, 82. 82 plus 18. How much it is? 100. 100 into 100. So that is equal to what? 18. So 18 is indicates what? The distance between these two genes is 18 centimorgan. 18 centimorgan. Are you followed? 18 centimorgan. So this is the distance between these two genes. This indicates what? Single crossover only it is. So with reference to 1 point only we can explain. So 1 point means here only exchange of one gene takes place in the crossing over. So with reference to single crossover, we can expect non-parental characters. The non-parental character combinations are always less than the parental combinations. That's why how many are with non-parental characters? 9 plus 9, 18. 18 by 100 into 100, we get in 18. Percentage of the recombinant percentage in between VG plus and B. So this 18% expressed in the map units, it is equivalent to 18 map units or 18 centimorgan. So here parental combination is more than 50%, recombinant less than 50%. Always the crossing over percent never exceed 50%. In CSR 2016, it is asked, one of the MCQ question asked in the CSR net. So crossing over, recombination of genes on the same chromosome is accomplished by come over a process by which parts of homologous chromosomes are interchanged. That is, exchange takes place. We already explained like this. So I will go for the next slide. This is about the crossing over formulas. Next, we can go for what is meant by interference. Interference means the tendency of one crossing over to interfere in the occurrence of another crossing over in the nearest vicinity or adjacent region. So, for example, if the chromosome, this is the chromosome, here there is a chance takes place for the crossing over. So what is meant by interference? The tendency of one crossing over to interfere in the occurrence of another crossing over. If any chromosome shows multiple crossovers, multiple crossover means any chromosome, the linked genes are changed or exchanged with each other by formation of chiasmata in more than 2 points or 3 points. This crossing over inhibits the occurrence of another crossing over near the vicinity on the same chromosome. Such type of phenomena is called as interference. Interference means the tendency of one crossing over to interfere in the occurrence of another crossing over in its nearest vicinity or adjacent region. So this phenomena of chiasma interference or chromosomal interference was first observed by H.J. Muller in 1916. So what is meant by these interference and what are the characteristics? The degree of interference may vary in different regions of the chromosome. Sometimes it will be inhibits, sometimes will be there is a partial favor for the one more crossing over in the nearest place or in the vicinity of the same chromosome. So it is found to be inverse proportionality to the distance between the points of crossing over. So it is found that inversely proportional to the distance between the points of crossing over. It means interference is maximum over a short distance and decreases as the distance increases. When the distance between the two genes increases, automatically it decreases the interference. So it is a maximum near centromere and telomere. Generally, the interference is maximum near the centromere and telomere region. In the practically, it is proved in Drosophila, interference prevents the double crossover between the genes of 10 to 15 units apart. So it is practically proved in the Drosophila, interference prevent the DCO. DCO means double crossovers between the genes of 10 to 15 units. So the degree of interference is measured by coefficient of coincidence. So this is one more formula related to the 3-point test cross. Coefficient of coincidence is equal to observed double crossovers by expected double crossovers. If any 3-point test cross problem, you can analyze the data. Definitely the minimum number of progeny in the entire data, in the entire total progeny, very less progeny number is there that indicates double crossovers. ఇచ్చిన ప్రాబ్లంలో ప్రాజనీలో అతి తక్కువ నంబర్ క్యారెక్టర్ కలిగినటువంటి సంఖ్య డబుల్ క్రాస్ ఓవర్ను తెలియజేస్తుంది అన్నమాట. సో దట్ ఈస్ నథింగ్ బట్ అబ్జర్వ్డ్ డబుల్ క్రాస్ ఓవర్ అవుతుంది. అది ఏమైతుంది? అబ్జర్వ్డ్ డబుల్ క్రాస్ ఓవర్స్. Sometimes the level of interference are given as the coefficient of interference also, which equal 1 minus coefficient of coincidence. That is equal to I is equal to 1 minus C antar. I means interference, 1 minus C. C means coefficient of coincidence. So what meant by positive interference? What meant by negative interference? Positive interference indicate that one crossing over interferes with the occurrence of second crossing over nearby. Interference value between 0 and 1 indicates positive interference. ఎప్పుడైనా ఇంటర్ఫరెన్స్ వాల్యూ 0కు 1కు మధ్య కనుక ఉన్నట్లయితే అది పాజిటివ్ ఇంటర్ఫరెన్స్ అవుతుంది. ఇట్ ఇండికేట్స్ పార్షల్ క్రాసింగ్ ఓవర్ జరగడానికి స్కోప్ ఉంటుంది. ఇంటర్ఫరెన్స్ ఈజ్ 100% అంటే అబ్సల్యూట్ 1 అయిందనుకో డబుల్ క్రాస్ ఓవర్ ఆర్ ఆల్ టుగెదర్ absent అంటే నో DCO అనమాట. ఇంటర్ఫరెన్స్ ఈజ్ ఈక్వలెంట్ టు 1. దెర్ ఈస్ నో ఎక్స్పెక్టేషన్ ఆఫ్ DCO in between the genes. That is double crossovers are not takes place. If the frequency of double crossover is equal to the expected value, it means that there is no interference and I is equal to 0. It is generally observed in eukaryotes. What is meant by negative interference? Negative interference indicates that the occurrence of one crossing over increases the likelihood of additional crossing over occurs in the nearest vicinity, so-called clustering of crossing overs. It occurs in certain microorganisms like bacteriophages and fungus only. So these are the things you can remember, the points: positive and negative interference. Okay. Next. So this is the characteristics. See here, coefficient of coincidence value here 0. If DCO is equal to 0%, that is absolute and 1 annamata ikkada. Less than 1, DCO less frequent than expected. Some partial interference is there. If coefficient of coincidence 1, the observed DCO is equal to expected DCO annamata. 1 minus 1, 0. అంటే interference none అవుతుంది అన్నమాట. I is equal to 1 minus C అని చెప్పినాం కదా. I is equal to 1 minus C అయినప్పుడు I is equal to 1 minus 1, 0. That means observed DCO is equal to expected DCO. That type of interference is none, అంటే 0. If the coefficient of coincident value greater than 1, we are getting negative value. That means DCO more frequent than expected. Expected దానికంటే ఎక్కువ మనం DCO అని అబ్జర్వ్ చేస్తాం. That indicates negative interference. Negative interference. So this table is also very, very important. Sometimes in the competitive examination, ask the relationship between the coefficient coincidence and type of interference. Okay. So now we can go for a problem, 3-point test cross problem. So for example, this is the 3-point test cross problem. In the Chinese prime rose, slate colored flowers, that is small s, is recessive to blue flowers. And ikkada blue flowers dominant, slate flowers అనేటివి recessive. Small s is indicates recessive. Capital S is indicates dominant. Red stigma, that is indicate gene R, small r, is recessive to green stigma, capital R. Green stigma is the dominant and red stigma is the recessive. In the same way, long style L, small l, is recessive to short style, that is capital L. All the genes were located in the same chromosome. The F1 of a cross between two breeding strains, when test crossed, gave the following progeny. Okay, F1 hybrid, no, recessive homozygous character తో ఉన్న ప్లాంట్ తోటి హైబ్రిడైజేషన్ చేసినప్పుడు మనకు ప్రాజనీ ఇట్లా వచ్చింది అనమాట. 8 type of progenies వస్తాయి ఇప్పుడు, ఎందుకంటే మనం 3 gametes వస్తాయి కాబట్టి based on the 3 gametes we can expect 8 type of progeny. Phenotypically we are getting 8 type of progenies are there. So based on the data we can solve this condition questions. What are the genotypes of the parents in the cross of the two breeding strains? Make a map of these genes showing gene order and the distance between them. Derive the coefficient of coincidence for interference between these genes. So first మనం డాటాను పరిశీలించినప్పుడు ఏం చేయాలంటే, ఇక్కడ ఉన్నటువంటి క్యారెక్టర్స్ తోటి ఆ ప్రాజనీ నంబర్ ఎంత ఉందనేది మనం ఇక్కడ గమనించాలి. సో బేస్డ్ ఆన్ దిస్ క్యారెక్టరిస్టిక్ మనకు తెలుస్తుంది ఏవి పేరెంటల్ టైపు, ఏవి క్రాస్ ఓవర్స్, ఏవి డబుల్ క్రాస్ ఓవర్స్ అనేవి మనం ఈజీగా తెలుసుకోవచ్చు. ఎట్లా తెలుసుకోవచ్చు అంటే, ఇన్ ద గివెన్ టోటల్ ఎంటైర్ ప్రాజనీ ఈజ్ 3000. అమాంగ్ ద 3000, highest number seen in the 4th one and 7th one. These 4th and 7th characteristics indicates that parental types. 4th and 7th. What is the 4th number is 977 and the 7th number is 960. You can say any, just now in the previous slide I can explain, the crossovers never exceed the 50%. And always crossovers అన్ని కూడా 50% లోపలనే ఉంటాయి. అంటే 51% parental types ఉంటే 49% recombinants ఉన్నట్టు లెక్క. That is a model example. But in this problem, so 977, 960 are the parental types. In the vice versa, very less number of progeny. See here 27 is there, again 27 is there. That is 1 and 8 indicates that double crossovers. 1 and 8 indicates that డబుల్ క్రాస్ ఓవర్. అంటే రికాంబినెంట్స్ లో డబుల్ క్రాస్ ఓవర్ ని ఎట్లా ఐడెంటిఫై చేస్తారు ప్రాబ్లమ్ లో అంటే, ఇచ్చిన ప్రాజనీలో ఏదైతే లీస్ట్ నంబర్ ఉంటుందో అవి డబుల్ క్రాస్ ఓవర్స్ అని గుర్తుపెట్టుకోవాలి మీరు డాటాలో. ఇన్ ద గివెన్ డాటా విచ్ ప్రాజనీ హ్యావింగ్ వెరీ లెస్ నంబర్, దట్ blindly we can say that is nothing but they are double crossovers. Is it clear? Next, in the remaining 2 and 6, one kind of crossover, that is single crossovers. Another 3 and 5, this one more crossing over takes place. In the 3-point test cross, single crossovers are 2, double crossovers are 1. So how we can calculate the recombinant percentage in between these genes? So based on the characters, you can write the genes. Slate flowers, what you write here? Yes, small s. Green stigma, what you write? Capital R. And short style, what is the short style? Capital L. So small s, capital R, capital L. That indicates double crossover. Here blue color, blue means capital S, red stigma, small r, and long style, that is indicates small l. So in this way you can write the gene representative genes right in this order. Based on that we can easily calculate the percentages. See here, next slide. So first solution, how to solve this problem. So I will explain. With the 3 gene pairs, 8 phenotypic classes are expected and 8 are observed. So the most frequent reciprocal pairs of classes phenotypes which are arise from non-crossover gametes. Whatever the higher number in the progeny is there, they are nothing but non-crossover gametes. They are directly coming from the non-crossover gametes. So the genotypes of the original parents, that is equivalent to the original parents. Here the 2 classes 4 and 7 are slate flower and red stigma long style, and blue flower green stigma and short style 977 and 960 respective numbers. We can add both the numbers that is indicate parental types. Okay, so in the F1 triple heterozygous parent, this generation must have been this type of gene. We can expect the order, not correctly, but 3 genes are present on the chromosome, but in which order it is arranged we don't know, but we can expect SRL oblique SR, small r, small s, small l. So the true breeding parents were like this, blue green short and slate red long. So in this way we can write the just gene order. Then we can go for the double crossovers. 1 and 8 indicates double crossover. Double crossover, how do we can calculate the percentage of the genes which represent the double crossover? Double crossovers divided by total progeny into 100. That is equal to how much? 1.8%. 1.8%. The gene pair that has changed its position relative to other 2 pairs. ఎక్కడైతే జీన్ పేర్లో పేరెంట్ జీన్ కి ఈ క్రాస్ ఓవర్ జరిగినటువంటి జీన్ లో ఎక్కడ తేడా వచ్చింది, ఏ క్యారెక్టర్ లో తేడా వచ్చింది అనేది మనం ఈజీగా ఐడెంటిఫై చేయొచ్చు. ఎందుకంటే ఆ జీన్ అనేది స్మాల్ లెటర్ పైకి క్యాపిటల్ లెటర్ అనేది ఎక్స్చేంజ్ అవుతుంది కాబట్టి. హియర్ ఎస్ అండ్ ఎస్ హ్యాస్ చేంజ్డ్ ఇట్స్ పొజిషన్, దేర్ ఫోర్ ద ఆర్డర్ ఆఫ్ ద జీన్ RSL and L. We can diagram the F1 test cross as follows. So test cross లో ఇట్లా మనం చూపిస్తాం genotypically. Capital RSL by small RSL cross with small RSL. The homologous chromosome also small RSL. So a single crossover takes place between these 2 genes R and S.
Sorry, if
The
possible, please
representative number
zoom it.
427 plus 402 divided by 3000 into 100. We are expecting that 27.63.
ఒకసారి జూమ్ చేయండి సార్, అది కొద్దిగా జూమ్ చేస్తే పెద్దగా కనబడుతుంది. కొద్దిగా
it's visible now.
ఏం కావట్లేదు. ఓకే ఓకే సార్, ప్రొసీడ్.
Okay.
Yes sir. Okay, clear.
So in this way we can calculate the distance between the 2 genes R and S the representative numbers given in the data 427 plus 402, 829 by 3000 into 100. We are calculate the distance between these R and S, 27.63, that indicates 21.63 centimorgan. Similarly, another crossing over between S and L, that represent numbers are 95 and 85 is equal to 180. 180 divided by 3000 into 100, that is 6%. So the map distance between R and S is single crossover plus double crossover. అంటే ఈ సింగిల్ క్రాస్ ఓవర్స్కి డబుల్ క్రాస్ ఓవర్ వాల్యూను యాడ్ చేయాలి. మనకు డబుల్ క్రాస్ ఓవర్ ఎంత వచ్చింది? 1.8%. సో 27.63 plus 1.8, 29.43. In the similar way, in another gene set, S and L, 6 plus 1.8, that indicate 7.8%. So this is the distance between the genes, or we can calculate it based on the percentage of the recombination frequency. So now we can go for map distance. So the map, genetic map, construction of the genetic map, R and S, how much it is distance? R and S we can calculate 27.63, దానికి డబుల్ క్రాస్ ఓవర్ కలిపితే మనకు 29.43 వచ్చింది. That is equal 29.4 here. And 7.8, it indicates S and L. So the correct order of the gene is RSL. So this construction of the genetic map. So the next we can go for coefficient of coincidence. Coefficient of coincidence formula what? Frequency of observed DCO by frequency of expected DCO. How can you calculate the expected DCO? Expected DCO, observed DCO. Observed DCO means it is given in the data, that is 1.8%. Percentage కాబట్టి by 100 వేస్తే 0.018 వస్తుంది. How can you calculate the expected DCO? Expected DCO is nothing but it is the single crossover region 1 into single crossover region 2 minus, is equal to ఎంత వచ్చిన value? 0.023. This is the value of single crossover percentage region 1. This is the value of single crossover region 2. 29.4 by 100 into 7.8 by 100, that is equal to 0.023. Now what is the formula? Coefficient of coincidence is equal to frequency of observed DCO. How much it is frequency of observed? 0.018. Expected what? 0.023. That is equal to 0.78. 0.78. So 0.78 వచ్చింది కాబట్టి coefficient of coincidence. Interference ఏమవుతుంది? I is equal to 1 minus 0.78 అవుతుంది. I is equal to 1 minus 0.78 అవుతుంది. అంటే పార్షియల్ ఇంటర్ఫెరెన్స్ ఉందన్నమాట దీంట్లో. నన్ కాదు, అబ్జల్యూట్ కాదు, పార్షియల్ ఇంటర్ఫెరెన్స్ ఉంటుంది కాబట్టి 1 minus 0.78, that indicates interference value. So in this way you can calculate the recombinant frequency. By the help of recombinant frequency we can calculate the distance of the genes, and after calculation of the distance between the two genes, we can go for coefficient of coincidence and as well as interference. In this way you can solve the problems based on the given data only. That's why 3-point test cross is very very important with reference to set, net, and as well as our sometimes small questions are asked in the interference formula or coefficient coincidence formula. In this way you can prepare more number of problems you have to practice. Definitely got a good information regarding the 3-point test cross. I think everybody understood. Okay, sir.
Thank you very much, sir. So it is very hot topic, but it is most important topic for all national level exams like CSIR net and other competitive exams, central exams, and also the life science exams, AP set and TG set examination. Definitely 2-point test class, 3-point test class, and interference, coincidence. These terms, terminology, and also the problems definitely were observed in the all examinations, any type of examinations, including in present scenario, the CUET and CPGET exam also definitely we are observed one or two questions. So
Yes.
that's why this session is very, very important, and it is also very not only in a single time or double time, whatever it may be, but your practice is most important. Not a Sir has given some of the examples, but whenever we are practicing, definitely we are achieving these problems very easily. Understanding is most important. At the same time, the formula is also most important. So observed frequencies and expected frequencies, uh, uh, which one is the most important? That is, we should understand our level best. So sir has given very clear picture regarding of this 3-point test class. Really wonderful, sir. Thank you very much for your presentation. So tomorrow we will meet same time. Thank you,
Okay,
sir.
sir. Thank you.